I'm a pathologist here at St Vincent's, and I'll just talk about, this is the Ocular Melanoma Summit, but I won't talk about eyelid. That's cutaneous, and that's been well addressed in Australia and internationally. I'll limit my talk to conjunctival and uveal, and I'll limit it to primary resection specimens because Daniel's covered the fine needle aspiration aspect quite well. So looking at conjunctival melanoma, and just looking at this from a very logistical pathology sort of workflow point of view, the samples are typically small, they're a few millimetres across. Sometimes the clinician doesn't suspect the pathology, especially if they're a general ophthalmologist. Fresh tumour banking is nearly impossible because they're quite small, and in our population people tend to present early, so we don't get great big conjunctival tumours. But we can perform molecular testing on FFPE, that's formalin fixed paraffin embedded tissue. So I'll show you any time we get a conjunctiva, and this happens to be a very big one, it's about 8 millimetres across. We always embed the entire specimen, even if it's not a melanoma, because we just don't know. That way we can assess every single margin all the way around. And this one's marked with a micro suture, so you can sort of see the suture here. So then we'll be able to orientate it for the surgeon as well. So there's at least 2 blocks so that we can assess all margins. All of the conjunctival specimens are fixed, and this surgeon has very kindly put the conjunctival melanoma on a piece of cardboard, and that flattens it out. Otherwise it'll curl and it'll be pretty much impossible to give you proper margins. So it's fixed and paraffin embedded, and that's just a standard thing that we do in histopathology. And the standard protocol is to get hematoxylin and eosin for morphology, a panel of immunohistochemistry, and then we can give the TNM staging according to AJCC from the pathology point of view. The paraffin block allows you also to do molecular testing, as I mentioned. And there's often limited tissue left. If you want to do, for example, making a tissue microarray from a conjunctival melanoma sample could be quite tricky, but getting additional unstained sections is probably possible depending on the size of the tumour. This is what a conjunctival melanoma, an early one, looks like. This is the conjunctival surface, and a lot of this disease is in situ, but we've got some melanoma cells starting to invade into the stroma. Here is another case where most of the disease is in the stroma, that's the conjunctival surface. And our panel, we usually have a protocol for conjunctival melanoma, which includes 2 melanocytic markers and BRAF. So we can actually have a look at the protein that's produced by this specific BRAF mutation to see whether it's there or not. It doesn't tell you anything about the other BRAF mutations. And this happens to be HMB45. In pigmented lesions like melanomas that are brown, we tend to use a chromogen that's red so that we can distinguish that from the brown that's in the background. Molecular testing, we're very fortunate. We partner with our sister organisation in Melbourne, and they've developed next generation sequencing panel for the cutaneous melanomas, and because conjunctival, as Max mentioned, has very similar molecular pathology to cutaneous, we're able just to get the hotspot panel done down there. It's very informative. We use FFPE, as we said, and for stage 3 and 4 melanomas, for Australian patients, we have a Medicare item number that we can access, and that can be bulk billed. So for many of our patients, that's a real issue, and that can be done with no out-of-pocket costs. That's the platform that they use. It's an ion torrent platform. It's the 50-gene hotspot panel. These are the genes involved. The ones highlighted in blue are clinically significant for cutaneous and mucosal or conjunctival melanoma. And I've highlighted GNA11 and GNAQ in here as well because it's a uveal marker, but also melanomas arising from blue nevi will also have mutations in those 2 genes. Max showed you a part of what one of the NGS reports looks like. So this is one that does have a BRAF mutation. It tells you the specific mutation, the amino acid substitution, and how frequent that particular change is in your tumour. And we can corroborate that with our BRAF immunohistochemistry because it happens to be that particular mutation. There's a real problem in Australia because our college doesn't actually train us for ocular pathology, and although they have ratified cancer protocols for everything else, they don't have it for ocular oncology. So there is no RCPA protocol for reporting conjunctival melanoma. So we've drafted one that basically incorporates AJCC fields as well as some UK and US College of Pathology proformas. There's a lot of data for each tumour, and I would love for this to be extracted into a collaborative registry, because I think those who are doing research know that the quality of the sample really relies on the clinical and the pathological annotation, so it's going to make your scientific observations just that much more meaningful. I've kind of been influenced a lot by the neuropathology people, so in neuro-oncology now, and I think most tumours are moving towards this, and there's an integrated diagnosis approach. So you give a morphological diagnosis, you give them up the margins and the AJCC stage, but you also give a molecular diagnosis. So we're moving towards that for conjunctival and uveal. Moving on to uveal melanoma, the uveal tract is a pigmented vascularized stroma that arises from neural crest, and it consists of iris, ciliary body, and the choroid. And when we get an eyeball, this is decades-old methodology. The most important thing for a pathologist is to orientate it. It's easy when it's in the patient, it's not so easy when it comes out. So if you look at this right eyeball from the back, this is the optic nerve stump. These are the rectus muscles, these four out here, and there's a superior oblique, an inferior oblique, and some vortex veins, and they're your landmarks. The old protocol actually used to take 64 hours before you even get to gross the eye. So that's a long time, and why did they do that? Well, it's because what we really want for staging is to get the central block, and that's the one that's in the middle. It's the pupil to optic nerve block, and that means you've got to trim the top and the bottom off, those little caps or calottes. And it's hard to take a calot off when the eye is not fixed. But it turns out that it's only hard to take both calots off, that you can actually take one calot off really easily when the eye is fresh. As long as you keep the rest of the eye intact, fix everything, nothing goes wrong with your protocol. So what we've decided to do in collaboration with Michael and with Max, who are the surgeons that have been referring to me so far, is we orientate the eye while it's fresh. We know that it's coming at least 24 hours ahead. We transilluminate it, measure it, and then we harvest the vortex veins, and then we remove one calot. So when we transilluminate, we know where the tumour is, and hopefully when we take one cap off, we'll catch some tumour in there. That leaves the rest of the eye, which we submit into formalin and fix that really well. It actually fixes faster because now it's opened. And from this calot that we've taken off fresh, there's usually plenty of tumour for both banking, molecular testing, and people who want to try other applications, such as Alex, who might want to try a single cell to suspend this. So we bank the tumour from this one, and we actually embed everything. Once the second half is fixed, what we do is we do some macro photography, and we can get some really good ideas about the tumour. In this particular case, it's moderately pigmented, it's sitting at the equator, extending to ciliary body and posteriorly. If you rotate the eye, this happens to be one that was at the temporal eye, and this is yellow spots, the macula lutea. We've got optic nerves, so it's almost a clinical view, and we can have a really good idea of what structures it's involving anteriorly and posteriorly, and we can give a really good measurement of its size. Then when we proceed to histology, we can tell all of the high-risk prognostic factors. This one happens to be epithelioid. It has all the closed extracellular vascular loop, so it's here's a mitosis. So those are all factors that are high risk. We also do BAP1 immunohistochemistry, so this gives us a surrogate, and it tells us if it's not being expressed by the tumour nuclei, there's likely to be biallelic inactivation of the BAP1 gene. So you have to ignore these clumpy cells. They're actually tumour-infiltrating macrophages, and the small ones are probably lymphocytes. The endothelial cells in the vessels will express on BAP1, but the tumour cell nuclei itself in this particular tumour was absent, and that correlates with an epithelioid morphology and typically monosome in chromosome 3. Again, we don't have a structured protocol that's ratified by our college, but I've been trying to integrate a uveal melanoma report with both the molecular findings and the histopathology, and I have this many data fields. So I would love for that to be extracted for all of these tumours. I think it could be informative. Our standard routine immunohistochemistry protocol will include CD31 for the vascular density, CD163 and 68 to look at the tumour-infiltrating macrophages. Of course, we do BAP1, MART1 just as a melanocytic marker, and just for a research look and see, and because Anthony really wanted it, we look at PRAME. I'll just show you another example. Uveal melanomas can be amelanotic. This one's quite white, as you can see, and it happened to be mostly spindle, so that's a lower prognostic histomorphology, and this is what BAP1 looks like in the lower lower risk group. So you can see really strong nuclear expression of BAP1 in all of the tumour. Sometimes, and often, the small uveal melanomas are rarely enucleated. As we heard from Claire, they're usually radiotherapy with plaque or other modalities. But this is Mike Giblin's patient. I dislodged that lens artifactually when I was sectioning, so that's why you're seeing it in the vitreous cavity. But this was a ciliary body and iris melanoma that was symptomatic, and the patient wanted out. So it's quite subtle, but there it is, a thickened ciliary body, and probably rotavirus is looking quite abnormal. That's what it looks like on a slide for us under the microscope. That's the H&E stain. And on higher power, we have the lenses here. This is the pars placata of the ciliary body, ciliary muscle. So we've got anterior ciliary body, there's tumour cells going into meshwork. That's cornea and iris. And on higher power again, you can see it's coming across more onto the iris here. Happened to be epithelioid, but it was so small that we couldn't actually, this is one setting where we couldn't get fresh tissue. It would have been quite hard to bank. We couldn't get our molecular test because ours is based on fresh tissue. But this is where our BAP1 was quite informative as well. Again, you've got to ignore the tumour-infiltrating macrophages and look at the tumour cells, and it's BAP1 deficient, so probably would correlate with monosomy as a surrogate marker. So looking at the molecular pathology of uveal melanoma, we've already had both Nick and Ellen tell you about that. But as you know, it's very different to mucocutaneous melanoma, and unlike conjunctival melanoma, we don't do molecular testing to look at therapy. We look at it for risk stratification, so it's just for prognostication at this point in time. We've known for decades, as Bertel told us, that disomy 3 tumours do better, monosomy tumours do worse. It turns out chromosomes 6 and 8 are quite important too. The platforms, you can just do conventional karyotyping and put fresh tumour cells and push them into metaphase and stain them with Giemsa and then document them all. You can use MLPA that Liverpool does, and when they have less than enough DNA, they can use a microsatellite test. You can do fluorescence in situ hybridization. Gene expression profiling, which is a proprietary test that you can do. It's quite expensive, about $7,000 US. Comparative genomic hybridization / single nucleotide polymorphism array, I'm just going to call it a SNP array, is another method where you can look at all the chromosomes, and next generation sequencing is more and more coming up as a diagnostic test that can be used. This is conventional karyotyping. You look at the morphology and count the number of chromosomes. This is not a typical karyotype for uveal melanoma. The laboratory that I work with is Dr. Lucas is here in the audience. She is the head of that lab, the cytogenetics lab, and she can do this, but it's a little fiddly and we've moved away from doing that. We can use fluorescence in situ hybridization, so you've got to use a fluorescence microscope. Usually have one probe that looks at a particular gene. When it hybridizes, there's a fluorescent marker. There's usually one centromere marking probe, which is green, that tells you that there's a chromosome there. And then in this particular instance, it's BAP1, which is on chromosome 3. So you count the number of chromosomes per nucleus, so all these blue things are nuclei, the number of BAP1 signals, and then you see whether it's lost or gained. So it's quite fiddly, and we usually do one probe per section. So if you wanted to look at 8, we're looking at the MYC gene on chromosome 8q. So we've moved away from that because it tends to be less informative, and it can be a little bit technically challenging when you're looking at more than one probe. Why we're using the SNP array, I think because we can get a whole of chromosome picture, we can get quite high resolution looks at multiple genes on each of the chromosomes. And there's handout out the back if you're interested in this particular test. Why do we want to use this? As Ellen and Nick showed you, that the Cancer Genome Atlas Consortium was able to subclassify uveal melanoma into 4 molecular groups, and they correlate very well to prognosis. So we can get that information from this test. We've also accredited with NATA. So in Australia and New Zealand, you can't offer a diagnostic test unless it's accredited by our accreditation body. There is a Medicare rebate that's attached to it, so that's really helpful for a lot of our public patients. And the surplus tumour cells can actually be used for in vitro assays, for single cell sequencing. Disadvantages, you do need fresh tissue and you do need a fair bit of it. And it's also not a mutational test. It doesn't tell you the sequence of the genes. So this is what Robin would grow up in culture before she goes and does the test, and this actually makes sure she dissociates the tumour cells and makes sure there's no lymphocytes, macrophages, stromal cells, and she's just getting pure tumour DNA, and there's often excess tissue after we did the SNP test. This is what the result is. So the raw data is here in the little pink dots, and you can use this to generate a picture of each chromosome. So that's chromosomes 1 to 22, and then the sex chromosomes, and then you can see where arms have been lost or gained. This has already been presented to you. So these are 80 resection samples from TCGA, and it's really spectacular science. It's a lot of technology. There's whole genome sequencing, there's exome sequencing, copy number alterations, mRNA, long non-coding RNA, microRNA, the whole methylome, the whole proteome. And if you put that all together in these 80 tumours and correlate it to survival and metastasis, 4 subgroups fall out. So one is the disomy group, one is the monosomy group. In the disomy group, these 2 genes in this particular set are mutually exclusive. So if you fall into group 1, you essentially have what is probably a nevus. Group 2, you may develop late metastases, and these are the poorer prognostic groups. So, very fancy stuff. How do we integrate it into our diagnostic workflow? How do we make use of it now and cheaply and readily for our patients? And I was really gobsmacked when I had a look at this particular figure in that paper, because if you look at the somatic copy number alterations, you can actually pull out those 4 subgroups just by looking at the status of chromosomes 3, 6, and 8. So you could pretty much do conventional karyotyping and get those risk stratification groups. I was pretty blown away. Because, we don't have whole genome sequencing validated to NATA standards anywhere in Australia. We don't have even next-gen sequencing that's validated. There's no Medicare item number to bill. Methylomic analysis is in its infancy in the diagnostic labs. So most labs will be able to do this if you have a cytogenetics lab. So some of you might be saying, well, that's only 80 tumours. Fair enough, it's rare, but, I'm not that convinced. And Robin drew my attention to this paper from the Shields. Came out a couple of weeks ago, 658 samples. And these were all brachytherapy samples, so they're small to medium size, not resection specimens. And they actually looked at the TCGA groups based on chromosomes 3, 6, and 8. And lo and behold, those 4 groups fell out, both in terms of metastases, late metastases, and survival. So now that this information is out there, and it seems really hard to argue with their sample size or their methodology, but I'd appreciate everybody else's viewpoints on this. I did wonder whether we should be stratifying our patients. So they came out with some pretty strong comments in their discussion, and they said that it can reliably predict metastases and death. It can identify patients who need more intensive surveillance. Can be employed for patient selections for trials. And it blew me away. They said that even when they adjusted for clinical tumour thickness and height, it was an independent predictor. So I'm loath to argue with the Shields, and they do have a very big data set, and this is now out there, so our patients will be able to find this information now. And so I leave you with some questions which hopefully we can open up for discussion at the end of today. And that is, should we adopt the standardised structured reporting for ocular melanomas in Australia and New Zealand? Should we offer molecular testing for conjunctival melanoma the way that we do for cutaneous, given the molecular pathology is similar? We've had a few children with conjunctival melanoma, and we wondered what the appropriate referral pathway for that could be, because they may have a germline melanoma syndrome. Do we offer all uveal patients prognostic testing? Do we adopt the TCGA4 risk category stratification system? And for young patients with uveal melanoma, when do we say they should be referred for BAP1 sequencing, just in case we can pick up a familial BAP1 syndrome? So just food for thought for the audience. I really want to acknowledge Dr. Lucas, who all of the molecular data I've presented it was produced by her laboratory. She's head of cytogenetics at SydPath. Max, who's a wonderful collaborator both clinically and in a research setting, and brought the MBT to this particular campus. Mike Giblin, who I call the godfather of ocular oncology in Sydney, and he's been really strongly supportive of us from the very beginning. Anthony Joshua, it's really hard to get anyone interested in ocular oncology because it's so small, and hospitals are really reluctant to allocate resources, but when you get a head of medical oncology who's interested, you're on a winner. You've got some cut through. And we're really excited to share that Sarah Kummerfeld and Luke Hessen, two scientists here at Garvan, are part of the team, brilliant scientists, and my colleagues who are, help me every day with the histopathology reporting, Peter Earls and Wade Barrett. Thanks.